Okay, so today we're going to be looking at the kind of movement from DNA to RNA and to protein. So there are three major parts to this: replication, transcription, translation. This video is only going to be focusing on replication. So replication, exactly what is it? It's how DNA is copied and how we get other copies of DNA, how it's reproduced, whether something's going through mitosis or anything like that. How do we get more DNA? So DNA, if you want to think about this, an easy way to think about it is as a zipper. So DNA, if you line it up side by side right here, it kind of looks like a zipper. So if you unzip it, you end up with some of your bases, your nucleotides on one side and some on the other. And so you end up with two opposite strands, but the nice thing is because of Chargaff's rules, where A pairs with T, C with G, and so on, as you move down, you can make a copy of this. And so you start with just one backbone, and you could end up with a replication of your original strand. So you end up with two copies of DNA. And this is something called semi-conservative replication. However, we didn't know about this for a while. We talked about in the last lecture about all the different discoveries of DNA. And this was something that still needed to be proven. The guys who did it were Meselson and Stahl. And there were three hypotheses they had. They said either DNA replication was semi-conservative, where you had each strand, two strands make up DNA, so each strand of that split off and that made its own copy, just like we talked about. Or it was conservative, where you had one strand, one DNA molecule going, and it would replicate and you'd get a copy of it. So you have the original and then a copy. The last one was dispersive, where you take chunks of DNA and they'd split up and you just fill in the gaps, is what they thought. So these guys were going through and they were doing these experiments, and what they started out with was a bacteria with nitrogen that was 15, a heavy isotope, and then bacteria that were just regular 14 nitrogen, which was our lighter isotope. And so they mixed these, they transferred them, and they let them replicate. And what they saw over time was that the first bacteria generation had 100% of this N15, right? That's the amount that we initially started with. Once they cultured it, once they transferred it to the 14N, then you started to see both were 50-50, right in between, at about 14.5. And that's something you could see if you were able to go down and look at their DNA. You'd see half of it had this heavy nitrogen, which is a big part of our nucleotide bases, is nitrogen. And then the other half was this 14. So over time, they let these things go and they had more and more generations divide. And what they eventually saw was a separate band started to form up at this 14. So you'd get one band at about 14.5 and the other band up at 14. And over subsequent generations, the percentage that were this one part kept increasing. Okay. And so what they came out with this, what their conclusions were that it wasn't a conservative model, because if you did that, you would expect two separate bands, one at the 14 and one at 15. And over time, we didn't see that. We didn't see this initial observation, we didn't see the second one either. If it was dispersive, you'd see everything at the 14.5, because you're just dividing this up into chunks. And then the subsequent generations, you'd see more and more of that 14.5. That's not what they saw. So what they came up with was this semi-conservative model, where you take it, you split the DNA, half of it has the 15, half has the 14. Over time, you get replication of that, and you end up with two distinct bands. You have a lot at 14, because these bands are going to go off and make the 14 weight. The other ones are at 15, so they're going to split off and go with 15. So there are three main steps for DNA replication, and we're going to look at a bunch of these different things. Again, it helps to think of this as kind of a zipper. So the way this starts is by looking at these things. It starts in what we call a replication bubble. So it splits DNA in two. Because it has two strands, it'll split them apart at these replication ports, pointed to with these pink arrows. And so we have a difference here between prokaryotes and eukaryotes. The prokaryotes have circular DNA, so they're going to form one replication bubble. It's just going to move around in that circle until they have two complete circles, and they're going to split off. It'll look something like this. Whereas eukaryotes, because it's a linear DNA, and we want to make this go as efficiently, as quickly as possible, we're going to have multiple replication bubbles happening here. And so these would be going in multiple spaces, like you can see here. We've got three of them in this one, rather than just one here. So the way all this works, the way this replication bubble even starts, is through the help of a bunch of different enzymes. So if we look at our enzymes here, think of DNA as a helix, and that helix is wound really tightly, and that's to protect the DNA. Now we need to unwrap that so that we can actually get access to the bases and do all this. The enzyme that's going to help us unwind and take away some of that torque, some of that pressure, is topoisomerase. It's the one that unwinds it and unscrews it a little bit. Next one, this kind of looks like a zipper to me. So I think of helicase as kind of the zipper. As you're pulling down, it's going to split those strands apart. So that's what this is doing. It's moving down and splitting these strands, as you can see here. Now, once you split these, you're going to have two separate strands of these DNA bases. Now, these aren't very stable. They want to bind to these. So to stabilize this, you have these single-strand binding proteins, which will go ahead and they'll allow DNA replication to occur. Also, when you're starting off with DNA polymerase, which is going to be the enzyme that goes through the replication of all this, you need a primer, an RNA primer to start it off. It's kind of like the key in the ignition, and this enzyme primase gives you that. So when you're doing DNA replication, it is autocatalytic. So DNA replication goes from the direction of 3' to 5'. So the 5', 3' has to do with the bases that are standing off of the molecules in the phosphate backbone. And so we have this anti-parallel. So it's going from 3' to 5', and it looks like this one is upside down. If that's how it's easy to think of it for you, that's fine. But this is just the phosphate backbone that will allow DNA to replicate. So once it starts off, once DNA polymerase starts this whole chain, it's just going to go. It's going to go, and it's going to add on these bases, whatever it needs here, whether it's T, C, G, it'll match up to. And this is just going to go and go and go over time. That's not to say there's not mistakes. We'll talk about that later. But it's autocatalytic. Once you start that train rolling, it's going to keep going. Okay? So once you've started DNA off, you've initiated it. Now you get to elongate it. So the way this is going to go is that you're adding nucleotides to this strand in 5' to 3' direction. But the problem, like I said, is DNA is anti-parallel. You have two strands. You have one 5', 3' going one way, one going the other. And so you're going to end up kind of running into this problem where you're going into the fork, which is an issue. So what's going to happen here is you're going to have this thing called a leading strand and a lagging strand. So let's take a look at the leading strand first. This is the easiest one to think of. So leading strand is just like you're going down and you have DNA polymerase binding. Once it's bound to that RNA primer, it's gone. It's that autocatalytic machinery just like we talked about. DNA polymerase is adding these bases, just chugging along down the tracks, kind of like a train. And as it's moving down, it's just opening and unzipping this DNA, and you leave behind these new DNA molecule, this new DNA base. Now this is opposite of lagging strands. So let's take a look at those. This is a little bit more complicated. So again, you need to move in this 5' to 3' direction, right? DNA is going to be going this way. So you start out with your RNA primer. You go for a little while with your DNA polymerase. This creates a thing called an Okazaki fragment, and this is named for the scientist that discovered it. And this Okazaki fragment is there, but it's not connected because you run into a problem. You run into this RNA primer again. And so you end up with a lot of little gaps like this all throughout this lagging strand. So it's called lagging strand because it takes a little bit more time for it to replicate. So you then have another DNA polymerase 1 comes in, and it adds in DNA where the RNA is. And then there's a little nick in here, and you have an enzyme called ligase, which comes in and it seals up that nick. So you end up with the same thing as you would for the leading strand, but it just takes a little bit more work to get to that point. So your leading strand and lagging strand. And if you look at this all together, you get a replication fork. So on one side you get your leading strand, and this one's just chugging along, going down the tracks, adding new bases. Then on the other side you have your lagging strand. Again, you have your primer, your primase, adding these RNA primers. You have DNA polymerase adding in, making Okazaki fragments, and then you're sealing it up with DNA polymerase 1 and DNA ligase. If you look at this all together, you've got all these different things working at once. So don't just think of them as separate things. If you look at it physiologically, it's all happening together. So there's a lot of machinery going on. And you guys can take a look at this a little bit. I would definitely encourage you to watch the videos because it's kind of hard to understand while I'm talking about it, but the videos do a pretty good job at showing it. The last step is termination. And so you've got, if you're in a prokaryote, you're just in a circle and that circle splits off, you're fine. That terminates it. But in eukaryotes, you have a little bit of a problem because it's longer and it's linear. You're going to end up with, when you remove these primers here and you're not making any more DNA, you have this kind of hangnail here, this little DNA hangnail. And this 3' end is over here and it's just hanging out. Now, this will be fine, it'll go, but what will happen is on that second round of replication, some of it, whatever pairs with this, is going to generate a shorter DNA molecule throughout this time. So as this moves on, this thing is going to get shorter and shorter daughter molecules, right? Because you end up with this same thing happening, this same process happening here, and then you lose all these bases. And if you keep going through this, you start to lose information. And this can cause a lot of mutations, a lot of things. So how do we kind of cope with this? Well, the one way that eukaryotes do it is with these things called telomeres. And you can see them highlighted here in this orangish red. And they are consistent lengths of DNA that repeat. So in vertebrates, it's TTA-GGG. And these are short repeating sequences that just signal, okay, this is the end of our DNA. This is where we need to stop. Okay, so there's not a lot of information gain there. It's just kind of a stopping point to prevent us from losing any information. The thing that adds on these things after elongation is done is telomerase. So this will add on all these TTA, GGGs, and that'll kind of signify the end of it. Okay, so that'll help us go through and prevent losing any information. So telomerase is also really important at the end. So I talked a little bit about these things just adding, right? They're just adding bases. They're gonna make mistakes. So we need to figure out how often they make mistakes, how do they fix these? So there are a lot of different polymerases that can do this. So they have a lot of different functions. One polymerase for here, we talked about DNA polymerase 3 is elongation in E. coli and other bacteria. In other eukaryotes, just DNA polymerase does the same thing, but it does it a little bit more slowly. So E. coli, this bacteria, does it at 500 bases per second. In eukaryotes, it'll do it at about 50 bases per second. Now, this error rate during elongation, like it says here, is about 1 in 10,000 mismatch bases. So think about all the bases of DNA, millions of these, 1 in 10,000, that actually becomes a pretty significant number. Right? So if we let this go, like it says here, you create over 300,000 mutations every single time a human cell divides. Now that's not good. Our cells will be completely wonky. We might get cancer much more quickly. So there is a way that this proofreads. These DNA polymerases will come in and they'll identify what is messed up. So they'll look at the bonding here. So you can see here these bonds are not correct. You'll have a nuclease that comes in and literally cuts out that chunk without disturbing the other pieces. You have another DNA polymerase coming in and fixing it. And then again, you have DNA ligase. It fixes up this little gap here and connects it all. So this is how it goes through. It'll just kind of read through, find some mistake, then it'll call in the enzymes to come in and repair it. So this is kind of the general process of DNA replications. It might take a little while for you guys to understand this, but it's just something that watching videos, going through it, kind of understanding the process of how it works will be very important, especially when you're talking about transcription, translation, and how this eventually becomes a protein. Okay, so like I said, go through this, look at it in a little bit more detail. I definitely watch the videos in here and see if you can identify what's happening. Okay, so that's it. Get ready for transcription and translation.